This video will complete our discussion on body fluids, particularly synovial fluid, semen analysis, and amniotic fluid. And thinking about synovial fluid is the fluid that surrounds your joint cavities or is within your joint cavities. So your shoulders, your elbows, your ankles, your knees, things of that nature. It's secreted by synoviocytes, which are the cells that line this area, and usually there's just a very small amount, it's 1 ml. But if something gets in there, if there's some sort of, degenerative process causing tissue trauma, some sort of infection, you can see an increase in synovial fluid. And you could almost assume that if you get a sample of synovial fluid in the lab, you're going to find something wrong, because you typically wouldn't go in and tap and take that fluid out through arthrocentesis unless something was up and the physician needed to see. There's not a lot to be taken if there isn't a disease thing at state. Currently happening in the patient. Synovial fluid can be analyzed for a few different things. You can sort of see how these different tubes might reflect different disorders. So you can have a disease that is a non-inflammatory disease. Letter A would most likely represent just like a normal synovial fluid, although the volume there is super high. So I'm wondering if that was actually synovial fluid to begin with. There can be inflammatory disorders, things that are causing fluid leakage into the joint capsule, and that would probably look more like letter B. Septic disorders would be either C or D, meaning you have some sort of infection going on in there. There can also be crystals, and this is going to be a really big one for synovial joints. We haven't really talked about crystal presence in the other areas of the body, but in the synovial fluid, if you get crystals in there, imagine sharp little needles in your joint capsule. It's going to be extremely painful to move that joint, and it's going to be causing a lot of damage. And you can see, again, letter D might accurately reflect that, because it's probably going to cause some trauma, which causes blood to show up. And then a hemorrhagic arthropathy. So there may be just bleeding straight into the joint capsule. All of these different shades and colors can vary, of course, but this is typically, the kinds of things we're looking for when we analyze synovial fluid. Typically, the synovial specimen is straw, viscous, ultrafiltrate. You can sort of see the bottom is more accurate for a normal specimen, and the top is something is off about it. It's way darker. It's kind of got a strange color to it. They would say that a normal synovial specimen is kind of like an uncooked egg white. It's very viscous, and that's because it does have hyaluronic acid in it. And hyaluronic acid, which we'll talk about in a minute, promotes viscosity in the specimen. As it says up here, it is an ultrafiltrate, so that means that it's basically plasma, but it doesn't have those big things, so big proteins and the big cells normally in normal conditions, and it normally does not clot. Now, if it does clot, then that probably means that you've had some introduction of blood into blood components into the synovial fluid. Fibrinogen is the protein that causes a clot, and if you have damage, it can sneak in and it can clot. So noting the presence of a clot is actually pretty important. The tubes, they don't necessarily have to be distributed in this order, but you will definitely have a sterile tube for microbiology, you'll have a hematology tube, and then some sort of plain tube to look for the presence of a clot. You could be doing other stuff too. This is not an exclusive list, but just kind of keep in mind, typically when you get a specimen like this, you would get something like that top specimen, and then it would be your job to distribute it out to the correct department. So it's not quite as mandated to have the first tube here, second tube here, third tube here, like it is for CSF, because just the nature of how you're going to receive it, it's all kind of going to be mixed in anyway. That hyaluronic acid, as I said, is something really unique to synovial fluid, and it makes the fluid very viscous and kind of stringy. When that property of stringiness is lost, that's a sign that something is up with our fluid. So we do what's called a string test, where you actually just pull some of the fluid up into, you kind of see a syringe, and you let it drop. And what should happen is that you should get about a 4 to 6 inch string before it breaks, if everything is okay. Now, if there's crystals in there, if there's inflammatory chemicals in there, if there's cellular material in there that's not supposed to be there, the string will break before it reaches 3 inches. If it does that, then that's a sign that something is up. So it means the hyaluronic acid is being I'll say impaired, but influenced by the other things that are in the synovial joint that aren't normally there. So it's literally just dropping it and looking to see how long that string forms. Seems kind of rudimentary, but it can tell you a lot. As I said, it's really important to notice the presence of crystals. We really haven't talked about that in other body fluids. We'll kind of talk about it a little bit in amniotic fluid, but crystals are a sign of gout, or what's called pseudogout. And this is basically when you build up uric acid or CPPD, which is just another form of crystal. You don't have to know what it stands for at this point. But these are two things that through metabolism can build up in your joints. And then when crystals build up in your joints, they kind of, I'll say, lock the joints. You basically can't like bend them because you have this deposition of crystals in the joint capsule. It's oftentimes very helpful to differentiate between if it's gout caused by uric acid. So uric acid is the crystal that causes gout, and CPPD is the crystal that causes what we call pseudogout. And the way that you can differentiate them is one, they sort of look different. So in the left-hand bottom corner, on the right is uric acid. It's very fine, sharp edges. And then on the left is CPPD. It's more like rhomboid, it doesn't have those big, long, sharp points. And then the other thing is if you have a really fancy microscope that can do this birefringence and polarized light, they are opposite each other when you add polarizing filters. On the left, you can see basically when the light is shining in the same plane as the crystal, it's either going to make the crystal look blue or it's going to make it look yellow. And CPPD, when the light is sort of shining in the same plane, and this is really grossly oversimplified, again, I'm not getting into the heavy physics of it, but when that light is in line with the crystal, essentially, CPPDs will look blue, and then if you compare that to the top right corner, uric acids will look yellow. And then if you adjust the light so that it's shining opposite that, or in a different direction, they actually sort of flip. And you can use that diagnostically to figure out which of the crystals you have, because sometimes it is hard to tell. I'm not going to expect you to do that. I mean, we don't even have those kind of scopes in our lab, but just know that crystals are really significant in synovial fluid, and then getting down to what kind they are is also really helpful from a diagnostic standpoint. The other body fluids that I'd like to talk about will be semen and amniotic fluid. And we are going to just briefly discuss semen analysis. It's actually quite an intense procedure in terms of analyzing sperm. And the reasons why we do semen analysis is one, for fertility or infertility issues. So if we have like a fertility specialist up at Aspirus Q&A, I know they have a pretty involved analysis lab up there because of that fertility specialist. In forensic cases, criminal cases of rape, things of that nature. Did the vasectomy work? You basically have to check to make sure there's no sperm in the ejaculate after vasectomy to know that it actually worked. And then how suitable is this semen sample for using for insemination, so an artificial insemination? There's many parts to analyzing semen, but these are kind of the reasons why the test is usually done. A semen sample needs to be processed immediately, and that's really the trend with all body fluids. They just have to go to the lab quickly because stuff stops working, levels start changing. I mean, it's similar to urine, but urine has a little bit of a longer shelf life. It has to stay at body temperature, so oftentimes people, when they have to bring the sample, they'll like stick it in their armpit to keep it at body temperature, because you have to look for the motility of the sperm, and if things cool down and the sperm essentially slow down and stop swimming, then you don't get to see it. So keeping it at body temp is really important. It's recommended that the person donating the sample is sexually abstinent for 3 to 5 days prior. And what will happen is, because that sample needs to get there really quickly, one of the reasons we need that so fast is because we're looking for liquefaction. Typically what happens is, shortly after ejaculation, the semen will coagulate. It has an enzyme that causes clotting, essentially. And from a technical standpoint, the reason for that is the sperm needs to stick to the cervix, which is the opening into the uterus, so that it can swim up the uterus and fallopian tubes and fertilize an egg. So it normally clots or clumps pretty quickly after ejaculation. It should liquefy again within 30 minutes, however. And if it does not do that, then that's a sign that something might be wrong. Or if it doesn't coagulate at all, that's also a sign that something could be up. So that liquefaction test is actually pretty important. The other thing that's really important is just looking at the sperm, observing them, and making sure that they're normal. So the tests that we're looking for, or the things we're looking at when we look at a sperm sample is, what does it look like? Are there any of these defects in the shape or the size? How is it swimming? Is it swimming in the right direction? Is it swimming in circles? Believe it or not, they can actually do that. Excuse me. What's the sperm concentration per milliliter? What's the total sperm count in the ejaculate? Are there white blood cells there? Yes, sometimes they're supposed to be, but there shouldn't be a ton. Is there infection? What's the pH? And what is the viscosity? So all of these things can affect fertility, right? And can affect the ability of sperm to fertilize eggs. So any one of them being off could be the cause of fertility issues. Speaking of fertility, moving right into amniotic fluid, here's the result of one of those sperm actually getting to the egg. And essentially, amniotic fluid is the fluid that is going to surround a developing embryo when it is in utero. So you can see in this diagram, there's like a water balloon basically surrounding the embryo. And as a result, this fluid, is bathing the baby. It's providing nutrients. It does a lot of stuff. It's pretty important. It's usually clear and colorless, but it can change color. Amniotic or amniocentesis is really risky because anytime you go poking into the bag that the baby's developing in, you run the risk of causing spontaneous delivery, or labor, I should say. And if you do it too early, then there could be issues. So going in to the amnion to get this fluid, you really have to have a good reason to do that. And there are good reasons. There are definitely good reasons, but it's not something that, you just happens because you want it to happen. Typically, there's risks. I mean, with any procedure, there's risks, but with this one, there is too. So if we suspect something called hemolytic disease of the fetus and newborn, basically the blood of the mom, the antibodies in the mom's blood are crossing the placenta and attacking the infant, you'll get into the amniotic fluid to see if that's happening. If there's bilirubin in the fluid, if there's blood in the fluid, if there's signs of inflammation in the fluid, that might be what's going on, and can be a fatal situation. So you will need to deal with that. Fetal lung maturity. There's all sorts of tests that we can do to see if baby's lungs are ready to come out into the world. The lungs are one of the last things to develop. And in premature infants, it's the thing that gives them the most trouble if they are born early, because their lungs don't have the chemicals, the integrity, what they need to actually inflate and breathe. And, they're put on little respirators or ventilators, I should say, and it's really dangerous. So oftentimes, if we know someone's going to go into labor early, they will draw the fluid so that they can tell if the baby's lungs are going to be ready to go or not. And depending on certain levels of things like sphingomyelin, not fibronectin, that's a different one, fibronectin, excuse me. But there's a whole bunch of things that can tell us if the lungs are ready. And if they're not ready, we need to be prepared for it if that baby's coming either way. The last thing I just wanted to talk about, it's kind of a test, but kind of not, is ferning. And if you look at the slide behind, or the background of this slide, basically, sometimes we've talked about the fact that you have to decide, is something urine or is it something else? And when a pregnant woman's water breaks, they oftentimes will either think they peed their pants, or maybe they did pee their pants and it's actually amniotic fluid. So what you do is, if you can get some of that sample, then you put it on a slide, you let it dry, and based on the composition of the amniotic fluid will actually crystallize, and it will look like it's like a fern. So that image behind are the crystals sort of spreading out and that ferning happening. And if you see that, that's a sign that yes, there's amniotic fluid in that sample that you received. You can read about the more specific amniotic fluid tests if you want. We're not going to go into it, but I want you to realize that it definitely is something that we can talk about. This will end our little discussion on body fluids. If you have questions, please let me know. You know how to find me.